Hello, you all, and welcome to the first of a couple of lectures on histology. So we're going to look at this briefly. We don't have a whole lot of time in this course, so we're going to take a brief sort of penciling through, see if you remember tissues and what they were all about. So when we classify all tissues of your body, no matter what tissue, they're all going to fall into only 4 categories, which is actually very little. It's either going to be an epithelial tissue, which means it's a coating, a covering, something that goes over something else. Or, let me skip the second one here, or it's going to be muscle, any kind of the 3 different types of muscles, or it's going to be a nerve, or the last one is the connective tissue, which is sort of the category of everything else. Everything else falls into that category. So what I'm going to look at now is epithelial and connective, and leave these other 2, the muscle and the neural, for later, because we're going to be doing a whole section on muscle and a whole section on nerves, and then we'll put in a little bit on the tissues. So let's start on the epithelial here. But first, I want you to get an idea of what goes on outside cells in tissues, because it's not just the tissue itself, but what sits around it. So in my picture here, I have a neuron, and it's colored beautifully. And then outside it, all that green stuff, what is that? You know, what's bathing a cell, essentially? You know, what is it? And we call it extra outside, cellular, outside of the cell, matrix. So it's what's floating around outside of these cells. And where does it come from and what is it? Well, it's actually synthesized by the cell itself. So it's the cell that makes its own surrounding. And what's it made of? It's glycoproteins, which is both a carbohydrate and a protein together, and all sorts of different protein fibers. So an example that you know really well is in the skin, and that's collagen, right? That gives that sort of filler inner of the skin. And there is elastin and all these different proteins that are going to be embedded in there. In fact, in the picture to the right here, there's all these long strands that you see in pink that are not really cells, but they are this sort of in between fibers. And then in between the fibers itself, all the lighter pink are all these different glycoproteins and the filler inners, I'm going to call it. So these are going to provide strength. They're going to be able to anchor a cell. So in this picture above here, you have cells sort of tied to each other. You have that matrix in blue in between. But we really do have to look also at how cells attach to each other and not like fall apart. How is it that you don't, you know, pinch your skin and end up with a piece of skin? So you have to find really good ways of attaching cells to each other if they're going to form a tissue. So I'm calling it glue. How to glue cells together in a tissue. And that's important because it'll be different in the different tissues. So there are 3 different kinds. There's tight junctions, desmosomes, and gap junctions. And we'll take a look at these, see if you remember all these junctions that put cells next to each other. So if you could put them all in one picture, it would look like this. This is really helpful. And in this diagram, you have a cell, another cell. See this little carpeting here made out of bunches of cells all put together. And we have some terminology that goes with that. This part that goes towards the lumen, which is the hollow part of an organ. So if you have an intestine, the hollow part is the lumen. That is the luminal membrane. And then the cell's going to go deeper, right? It's 3-dimensional. And on the base here is the basolateral membrane. So that's just, you know, are you talking about a cell that is facing the inside of the hollow part, or is it facing outside, essentially? But then next on this diagram is how do they stick so that they don't fall apart? So here there's a picture of this cell, cell 1. Let's do cell 1 and cell 2. And here, there's no passage. Nothing can cross here between these cells because these are kind of tied together there. So the question is, how do you tie them together? This is a more comprehensive diagram that has all sorts of tying together. So cell, cell, cell. And you have here, these are kind of like zipped together, right? Like a ziplock or a zipper. Get myself out of the way. And then there's other forms in which they connect. For example, here, this is not so much like a zipper, but like a clip. It's like it's pinched together there. Okay, and if you enlarge that, it looks like this with these funny little anchors. And then a third type is also here between one cell and another. But these actually look very different. Look, it looks like a little canal. It looks like it's a way of one cell to talk to another. Okay, so zipper and clip and little tubes that can connect. Okay, that's not what they're called. We're going to give you the real words. So tight junctions are the ones that I referred to as a zipper. They're able to glue cells really tightly. You want some cells to not be leaky. You can't have things leaking between the cells. And so they're going to have some sort of protein attachment, which really sticks them. So it's kind of, they're almost fused. It's a fusion of one cell next to the other one. And those proteins are going to interlock. And then there isn't anything that's going to leak there in between, right? So these are tight junctions. What's that going to do? It's going to block things. It's going to block things like water and solids. You don't have leaky things coming through here. So that makes sense. Where would you put that kind? And you would put it then in something like the digestive system. If you have water and bagel and burrito going through there and you have all this wet mixture, you don't want it oozing out into your intestine, right? So very important that these cells of your intestine are zipped. Tight junctions. This is what they would look like, we imagine. I mean, we've taken photos of this with scanning electron micrographs, but this is kind of the zipping in which in various locations these are sealed together, okay, all by proteins. The second one then is the clip-like, is the desmosome. So desmosomes are more like a clip than a zipper. They hold cells together. They're common in places like the epidermis. Okay, here, you don't have a whole lot of water that's trying to leak through, right? And, I mean, if you think of swimming, you have many, many, many layers on the epidermis. But what really has to happen in the epidermis and the heart is it has to be able to move in quite a few directions, right? The heart is always, you know, beating. And your skin, I can, again, take my skin and twist it, and it has to hold on. So it's important that you have these attachments between these cells. Very strong. Okay, they're strong. There is a stretching. There is this twisting. And when under scanning electron micrograph, they look like a J hook. So this is hooking onto the other cell. Okay, not so much zipped, but hooked here. This is a really nice picture of it, sort of how we try and show it to us people who are not looking under the microscope. And it would have this hook that connects to, this is one cell, right? And the pink here is one cell, and on the other side is the other cell. And here are these protein hooks that kind of sew it together in that one little spot. Okay? And in between, we call that just cement. But this is just kind of sewing two together with these protein hooks. And the last one is a gap junction. So I made a picture of a tube there because that's kind of what it looks like, right? You are going to allow small molecules to actually cross from one place to the other. It's not so much an anchor, it's a little bit of an anchor, but it's something that's going to allow communication between those two cells. It's very regulated, not everything can go through. So this is in cardiac and smooth muscle. Cardiac, very important, because when you have an electrical wave going through the heart telling it contract, right, contract, lub dub, lub dub, lub dub, then you need that electrical activity to be radiating through the cells. Excuse me. And how does it radiate? Through these openings between cells. So it could send that message very quickly. In smooth muscle in the gut, they also do exist. And this is interesting. One of the diseases is celiac disease. these gap junctions can be defective and allow too much water. Too much water going from place to place. This is one of the symptoms of a celiac, might have diarrhea, or exactly the opposite, when they don't let enough water in. So it's the defective movement of that water through these gap junctions. They could be too big, or they could remain closed. Very interesting how such a tiny thing connecting one cell to another can affect the whole individual so profoundly. So this is a sort of view where you can see that. You can see that little tube here, and opened, it's passing, right? You could pass something from cell 1 over to cell 2. They actually twist closed, and then nothing would pass. They're embedded proteins. Again, there are always proteins that are in between there. Let's throw some clickers at you. So if you had something like this, and I was showing you, oh, look at this connection between cells, and it looks like this, right? Where you have lots of sewing along there, then what would you call it? Not zipper connections. It's not alternative C. I'm going to skip over and let you figure that out. This next one would be another one where there's actually openings. This is a nice picture here where it shows it open or twisted closed, right? And then there's a passage from one to another. What would that be called? Button junctions, again, is not correct, and I will let you figure it out. The last one here is the one that's connected kind of with the J hooks. Hairy junctions is my choice for C. C seems to be the off-the-wall one every time. Okay, so I'm going to stop this video there. I'll do a next one then on when we actually start on the 2 tissues that I'm going to go over. So thanks for listening. Great. See you soon on the next video. Bye.